Welcome to Neuro Insights, the podcast delving into the frontiers of brain science and neurodegeneration. I'm your host, Rittika Dutta, and today we are unpacking the intricate world of protein misfolding and aggregation, a hallmark of Alzheimer's disease that's reshaping our understanding of cognitive decline. Our guest today is Kanak, who holds a Master of Science degree in Biotechnology and brings fresh perspective from her expertise in molecular biology. Kanak, thank you for joining us. To start, could you explain the fundamental biophysical processes behind protein misfolding in Alzheimer's, and particularly how amyloid-beta and tau proteins transition from soluble forms to toxic aggregates under cellular stress?
Yeah. First of all, thank you so much for having me in the podcast, Ritika. To start with the protein misfolding, it is a complex process that plays a critical role in the development of Alzheimer's disease. At the heart of this process, there are two key proteins. First is the amyloid beta, and second is the tau protein. They show the transitioning from the soluble forms to toxic aggregates under several cellular stress. So the amyloid beta shifts from alpha-helical to beta sheet structures, forming the fibrils that deposit as plaques, whereas the tau becomes the hyperphosphorylated, detaching from microtubules and aggregating into neurofibrillary tangles. These aggregates disrupt the normal cellular function, leading to neuronal damage. And I guess understanding these biophysical mechanisms is the key to developing effective Alzheimer's therapeutics.
That's a compelling overview. Actually, let's dive deeper into the triggers. Recent studies highlight the role of post-translational modifications like phosphorylation, ubiquitination in accelerated misfolding. And they indeed do accelerate misfolding. That is what the notion is. However, how do these misfolding interact with cellular chaperones such as heat shock proteins, and what cutting edge research suggests about their dysregulation in early stage Alzheimer's pathogenesis?
Yeah.
Yeah. Mhm.
Okay, so if we talk about the post-translational modifications like phosphorylation and ubiquitination, normally cellular chaperones like heat shock proteins recognize these modified proteins and assist in refolding them or targeting them for degradation. However, recent research shows that excessive or aberrant phosphorylation and ubiquitination can mask these chaperones recognition sites or overload proteostasis pathways, leading to toxic aggregate accumulation. Also, the cutting edge studies using the single molecule imaging, cryo-electron microscopy, and proteomic profiling revealed that in early Alzheimer's, these chaperones networks are dysregulated and HSP activity is impaired. Also, the co-chaperones are depleted and ubiquitin proteasome efficiency declines, creating a permissive environment for pathological aggregation.
Fascinating insights. Now, considering the Prion-like propagation hypothesis, the concept of Prion-like spreading of misfolded protein across brain regions is gaining traction. Can you discuss how seeding and templating such mechanisms facilitate this in Alzheimer's, and what advanced imaging techniques like, let's say, cryo-electron tomography reveal about the structural diversity of these aggregates?
Absolutely. So, in enzymers, the prion-like model suggests that once a protein like tau or amyloid beta misfolds, it can act as a seed, means inducing nearby, induce the nearby normal proteins to adopt the same misfolded structure. This templating mechanism helps to explain how pathology spreads in a stereotypical pattern across connected brain regions. Also, advanced tools like cryo-electron tomography now let us visualize these aggregates in 3D within their cellular context. But what's striking is the structural diversity they have. These fibrils aren't uniform but exist as distinct strains, each with unique conformations that may underlie differences in toxicity, progression rate, and clinical presentation. This level of structural detail is giving us a much clearer picture of why Alzheimer's behaves like a spreading network disease rather than a localized proteopathy.
Right, that's an excellent point actually. So let's talk about therapeutic implications right now. So antibody-based therapies targeting misfolded proteins have shown mixed results in clinical trials, which is what about news that has been coming out quite often. So, can we go deeper into the nuances into aggregate polymorphism and strain specific conformations that are emerging from single particle analysis, and how might they inform next generation immunotherapy?
Sure. So the single particle study shows that the tau or amyloid-beta exist as multiple strains with distinct structural folds. These differences expose or hide antibody targets, which explains why some therapies clear plaques but miss the toxic oligomers driving diseases. The next generation approaches are now aiming for selective antibodies, cocktails against multiple strains, and also the engineered Fc regions for safer microglial engagement. Matching therapy to aggregate structure is the key role here.
Right. Let's talk about genetic factors, which I think is the very next thing that we should talk about in coherence to this logic. So, like mutations in genes like APP, PSIN1, PSIN2 exacerbate misfolding, and that is a known fact. So, my question is, how do genome editing tools like CRISPR-Cas9, which has gained a lot of impetus, uh, help model these in organoids, and what epigenetic layers such as histone acetylation add to the complexity of aggregation pathway?
So, if we talk about the CRISPR-Cas9, that has been transformative role here. By introducing or correcting APP and other mutations in iPSC-derived brain organoids, we can recreate the exact biochemical shifts like altered amyloid beta 42 to 40 ratios and changes in gamma secretase processing that drive early Alzheimer's pathology. Also, this gives us a human 3D model to watch misfolding and spreading in real time. But it's not just the DNA sequence that matters. The epigenetic, as we know, the epigenetic landscape adds another layer into it. Like the histone acetylation, for instance, regulates expression of chaperones, uh, proteasome components, and autophagy-related genes. If acetylation patterns are disrupted, the cell's defense against these aggregation weakens a lot. By combining CRISPR editing with epigenetic modulation, we are uncovering how genetic seeds interact with an epigenetic environment that can either buffer or accelerate the aggregation pathways.
That's pretty intriguing, actually. So, now on environmental influences. So, environmental toxins and microbiome dysbiosis are linked with protein misfolding. What cutting-edge metabolomics data suggests about gut-brain axis contributions to Alzheimer's aggregates? And how does this lead to novel preventive strategies?
So, the metabolomics has given us the powerful insight into how the gut brain axis shapes the protein misfolding. And we are seeing that microbial metabolites like short chain fatty acids and bile acid derivatives can alter neuronal proteostasis and even promote the amyloid aggregation when dysregulated. Dysbiosis also brings in microbial amyloids that can cross seed with human proteins. Also, the exciting part is that this opens the door to primitive and preventive strategies by restoring a healthy microbiome through diet, uh, probiotics, or metabolite targeted therapies. We could reduce the biochemical signals that fuel these kind of aggregations, I guess.
Last layer, I think, let's discuss about AI. So integrating AI in predicting misfolding trajectory is, I guess, a very revolutionary idea. So how does machine learning models trained on molecular dynamic simulation forecast aggregate toxicity, and what interdisciplinary challenges remain in translating these personalized medicine for Alzheimer's?
So, if we talk about the AI models that trained on molecular dynamics simulations, can now predict how proteins misfold and which aggregate conformations are most toxic. This gives us a roadmap for anticipating disease trajectories before pathology fully sets in. And I think the challenge is integration, which is the combining structural data, genetics, and patient variability into models that are clinically actionable. And it's an exciting frontier, but it requires a lot tight collaboration between biologists, data scientists, and clinicians to bring personalized medication into practice for the models.
Kanak, you've truly illuminated the depths of protein misfolding in Alzheimer's. Thank you so much for coming to our show. Listeners, join us next for more on neurodegeneration. This is Rittika Dutta signing off.